Good morning. Today's topic is gene to protein. The process of formation of protein from a gene is known as gene expression. Actually, the information which is present in the DNA in the form of nucleotides, which is written in 4 bases, that is A, G, T, C, directs the formation of amino acids. That means the information present on DNA will be directed to form a chain of amino acids. So the assembly of this amino acid should be in the correct order to produce a proper functional protein. The DNA is inside the nucleus, as you all know, on chromosomes. The machinery of producing protein is outside the nucleus. So here information present on DNA is to reach outside in the cytoplasm, and there the informations are directed to synthesize proteins. There are 2 important steps in the formation of protein from a gene. The first step is transcription. Information from the DNA is used to make a corresponding RNA. The RNA which contain information to make a protein is also known as mRNA. mRNA is messenger RNA. The next step is, by taking information from mRNA or by using information from mRNA, proper protein is synthesized, and the process is known as translation. In short, DNA to RNA, then to protein. This is known as gene expression, or it is also known as central dogma of life. In prokaryotes, information from DNA is transferred to RNA. The process is transcription. This mRNA then immediately in contact with ribosome and forms polypeptide. There is no nuclear membrane, so from the DNA information could be directly formed to mRNA. This mRNA could directly meet ribosome and do the protein synthesis, that is translation. So DNA to RNA, then protein. This process takes place immediately. In an eukaryotic cell, we can see nucleus and nuclear membrane. So genetic material is inside the nucleus, whereas ribosome is situated in the cytoplasm. The transcription process, where information from DNA is used to make mRNA, the process happens inside the nucleus. The mRNA comes outside to the cytoplasm, and in the cytoplasm the process translation takes place. So unlike prokaryotes, in eukaryotes there is little bit delay. At first they are synthesized inside the nucleus, and that has to be transported outside to the cytoplasm, and you can see the translation happens outside the nucleus. Now one more thing which is to be discussed in eukaryote is, at first the information are copied to an RNA. This RNA is known as pre-RNA. As we all know, there are introns in the coding sequences of eukaryotic gene. These introns need to be removed. So you can see, after forming the RNA, which is known as pre-RNA, this pre-RNA is undergoing certain modification to make mRNA. So these modifications are known as post-transcriptional modification. So post-transcriptional modifications lead to formation of proper mRNA. Now this mRNA will transfer to the cytoplasm, and there it will be undergoing translation. The RNA, which act as a carrier of the genetic information from its site of synthesis, that is nucleus, and to the site of protein synthesis. Okay, so that is cytoplasm. That's why the mRNA or RNA is known as messenger RNA. They carry information, genetic information, from the site of its synthesis to the site of protein synthesis. The use of mRNA allows the cell to separate information storage from information utilization. See, the informations are stored in nucleus and informations are utilized in cytoplasm. The gene information can be amplified by having many copies of mRNA. So, from a gene, many copies of mRNA can be made. And these mRNA can be sent outside, and so you can amplify or increase level of expression of that gene by making many copies of mRNA from a single DNA. So it also gives the cell a greater opportunity to control both the level and the timing of expression of various genes. Once an mRNA is synthesized as a complementary copy of one strand of DNA, it moves into the cytoplasm and serve as a template for synthesis of protein by a process known as translation. This is general for all the living organisms. The central dogma for all the living organisms is DNA to RNA, then to protein. But certain viruses, which are known as retroviruses, they have their genetic material RNA. So first their information from RNA is copied to a DNA. This is known as cDNA. C means complementary. And from them again mRNA is produced, and that lead to protein formation. So there is a change in retroviruses. The process of formation of RNA from DNA is known as transcription. RNA to protein is known as translation. So in retrovirus, DNA is made from RNA. So this process is known as reverse transcription. Okay. So that's all regarding DNA to protein. Thank you.